Welcome to Healthy Aging and Beyond. Our brain is protected by a highly selective barrier, known as the blood-brain barrier, or BBB. This barrier is essential for keeping harmful substances out of the brain, while allowing vital nutrients to pass through. However, as we age, the integrity of this barrier declines, leading to increased permeability and a host of neurological issues. Recent research has uncovered a key factor in this process: senescence, particularly in brain endothelial cells, which are crucial components of the BBB. The blood-brain barrier is a complex structure composed of brain endothelial cells, pericytes, astrocytes, neurons, and extracellular matrix. Together, these elements form the neurovascular unit, which serves as a physical and metabolic shield for the brain. The endothelial cells lining the blood vessels in the brain are tightly packed together, creating a barrier that is both selective and protective. But this barrier doesn't stay robust forever. With age, the BBB becomes more permeable, allowing substances that should be kept out to seep into the brain. This deterioration is particularly pronounced in regions like the hippocampus, which is crucial for memory and cognition. The loss of tight junction proteins, altered transport systems, and reduced pericyte coverage all contribute to this decline. At the heart of this problem is cellular senescence, a state where cells stop dividing and undergo significant metabolic and epigenetic changes. Senescent cells accumulate in various tissues during aging, including the brain, where they can disrupt normal function. In the context of the blood-brain barrier, senescence in brain endothelial cells has been identified as a key driver of its dysfunction. In a recent study, researchers investigated how senescence affects the blood-brain barrier by analyzing brain sections from young and aged mice. They found that aged brain endothelial cells exhibited markers of senescence, such as increased levels of P21 and P16, which are associated with cell cycle arrest. These senescent cells also showed decreased expression of crucial tight junction proteins like occludin, leading to a compromised barrier. To delve deeper into the role of senescence, the researchers used a genetically modified mouse model known as INC-ATIC, where senescent cells can be selectively eliminated. By treating aged mice with a drug that induces the death of these senescent cells, they observed a significant improvement in blood-brain barrier integrity. The permeability of the blood-brain barrier decreased, as evidenced by reduced leakage of fluorescently labeled serum albumin into the brain tissue. This experiment highlighted the impact of senescent cells on blood-brain barrier function. Removing these cells not only improved the barrier's structure, but also restored some of its critical functions, suggesting that targeting senescence could be a promising strategy to maintain blood-brain barrier integrity in aging populations. The researchers didn't stop at mouse models. They developed an in vitro human blood-brain barrier model using endothelial cells derived from human cord blood. By inducing senescence in these cells through gamma radiation, they were able to mimic the aging process observed in the brain's blood vessels. The results were consistent with the findings in mice. The senescent human blood-brain barrier model showed reduced levels of occludin, leading to increased paracellular permeability, a measure of how easily substances can pass between cells. Additionally, the model exhibited lower transendothelial electrical resistance, indicating a weaker barrier. These findings confirm that senescence significantly disrupts blood-brain barrier function even in human cells. The implications of this research are profound. The breakdown of the blood-brain barrier is a critical factor in the development of neurodegenerative diseases like Alzheimer's and Parkinson's. By understanding the role of senescence in this process, we open up new avenues for therapeutic interventions aimed at preserving BBB function and potentially slowing down or preventing these debilitating conditions. One promising approach is the use of senolytics, drugs that selectively eliminate senescent cells. The study's findings suggest that senolytics could be used to strengthen the BBB in aging individuals, thereby protecting the brain from harmful substances and reducing the risk of cognitive decline. In conclusion, this study sheds light on the critical role that senescence plays in the aging brain, particularly in the breakdown of the blood-brain barrier. As we continue to explore the biology of aging, such insights will be invaluable in developing strategies to maintain brain health and combat neurodegenerative diseases. Thank you for watching, and if you found this video informative, don't forget to like, share, and subscribe for more updates on the latest in aging research.